Bismillahirrahmanirrahim. In previous lecture we discussed the classical pathway of complement activation. In today's lecture we are going to look at the lectin pathway of complement activations and alternative pathway of complement activation. Lectin pathway of complement activation. The lectin pathway, it involves C2 and C4 components of the complement system. It also involves the MBL, that is menin binding lectin, and picoline molecule. The MBL, it is going to binds to mannose, residues expressed by many microbes, and picoline is going to binds to acetylated carbohydrate. So these are the components involved in lectin pathway. Now we are going to look at the activators. Activators of the lectin pathway. The activators of the lectin pathway, it includes terminal mannose, which is a part of cell wall of many bacteria, fungi, etc. So when there is a pathogen in the body, it expressed the terminal mannose residues on its cell wall. So the MBL, which is found in blood circulation, is going to attach to these terminal mannose on the surface of pathogen, and the lectin pathway is going to activate it. The second activator of the lectin pathway, it includes acetylated or neutral carbohydrates. And these acetylated or neutral carbohydrates, it is also expressed by many bacteria. So these acetylated or neutral carbohydrates, for example, 1,3-beta D-glucan, it is going to activate the lectin pathway. To these acetylated neutral carbohydrate, the picoline molecule, which is a component of the lectin pathway, is going to binds to these acetylated or neutral carbohydrate, and as a result the lectin pathway is activated. So this is another activator of the lectin pathway. Another activator is activated PAMPs. PAMPs, it is a pathogen associated molecular pattern, and these PAMP we have already discussed in pattern recognition mechanism in our previous lectures. So these activated PAMP, it is also going to activate lectin pathway. Lectin pathway. So these are the activators of the lectin pathway. Now the steps involved in lectin pathway. The first step is binding of MBL or feculin to the surface of pathogen. Now remember that these MBL and feculin molecule, they are found in complex form with serum protease. Complexed with serum protease in blood, which we call, and these complexes are called mannose associated serum protease, MASP. So these MASP is going to attach to the surface of pathogen. Once these MASPs attach to the surface of pathogen, one of these MASPs, such as MASP1 or MASP2, they are going to cleave the C2 and C4 component of the complement system. And as a result of these cleavage, C4B2A complex is formed. And these complex, it is called C3 convertase of lectin pathway, and it is also going to cleave next component of the complement system, that is C3. Cleavage of C3 component. Clear? So what happens actually? The MBL in feculin, it is found in blood in complex form with serum protease, which we call the MASPs, mannose associated serum protease. And when these mannose associated serum protease, it attaches or binds to the surface of pathogens, one of these MASPs, that is such as MASP1 or MASP2, it is going to cleave the C2 and C4 component. C2 is cleaved into C2A and C2B. The C2A is a larger fragment and C2B is smaller. The C2B is released into fluid phase, and the C2A, it remains attached to the surface of pathogen. And C4B, it is also cleave into two fragments, that is C4A and C4B. C4A, which is a smaller fragment, it is going to diffuse out of the site of activation, and the C4B remains attached to the surface of pathogens, and as a result the C4B2A complex is formed, and this C4B2A complex is called the C3 convertase of lectin pathway, and this C3 convertase is going to cleave the next component of the complement system, that is C3. So it was all about the lectin pathway of complement activation. Now we are going to look at the third pathway of complement activation, that is alternative pathway. The alternative pathway, it is activated in the absence of specific antibody. So alternative pathway is a key part of the innate immune responses. Clear? It is non-specific, and the components involved in alternative pathway is the C3b component. Clear? This C3b component, it is found It is produced in blood by cleavage of a reactive group, a reactive thiol group of C3 component. So in blood, the C3 component is constantly cleaved into C3b fragments, and these C3b is responsible for the activation of alternative pathway. So the C3b is the main activator of alternative pathway. Or main component of the complement pathway. The alternative pathway, it can be activated by any type of foreign particles or foreign substances. The activator include lipopolysaccharides, and these lipopolysaccharides, they are expressed by is a part of cell wall of gram-negative bacteria. Clear? So whenever there is a lipopolysaccharide in the body, which is actually a part of the cell wall of bacteria, is going to activate the alternative pathway. The next activator includes cobra venom. Cobra venom, it is protein produced by some snake, and this cobra venom is also responsible for the activation of alternative pathway. It is protein produced by some snakes. The next activator, like classical pathway, some viruses, necrotic cells, and aggregated immunoglobulins. They are also going to activate the alternative pathway. In this alternative pathway, it is always on. Why? Because there is a factor in blood serum which we called the factor P, and these factor P, which is a serum proteins, and it is also called properdin protein. This factor P is responsible for stabilization of C3bb complex, which is a C3 convertase of the alternative pathway. And these C3bb, once it is formed, it is stabilized by factor P, which is found in serum protein, and as a result of this stabilization, the C3bb is going to cleave further C3 component. As a result of these cleavage, large number of C3b fragments are formed, and this is called, this huge wealth of C3b fragments is called amplification loop for alternative activation, pathway activation. And this is why the alternative pathway is always on. Clear? Due to this amplification loop. Just continuously the C3BB complex is going to cleave the C3 components, and C3B fragments are formed, and these C3B is going to attach to the surface of pathogens. So it is always on. And one thing more, that this preformed C3B, it can attach to proteins or carbohydrates, which are expressed by both, expressed by either host cell or bacterial cell or pathogen, you can say. So this C3B, it can attach to both the surface of host cell and pathogens. Clear? So its continual activations can damage the host cell, that is mammalian cells, but fortunately the mammalian cells has a regulatory mechanism. Clear? And these regulatory mechanism balanced the amplification loop for activation of alternative pathway under normal conditions. So it prevents the progressions of alternative pathway under normal conditions. However, the microbial cells, they lack these mechanisms and they cannot prevent the alternative pathway. So it leads to the subsequent steps of complement activation, and in this way the microbes are killed by alternative pathway. So these are the activator. Now we are going to look at the steps, all the steps of alternative pathway. Early steps of alternative pathway. Now the alternative pathway, it is activated by deposition of C3B component of the complement system on the surface of pathogen. So once the C3B component binds to the surface of pathogens, the alternative pathway is activated. Now there is another factor in blood serum called factor B. This factor B, it also binds to C3B, and as a result C3BB complex is formed. Clear? Next, in blood serum there is another factor called factor D. This factor D is going to binds to C3BB complex and cleave the B component into two fragments, that is BB and BA. The BA, which is a smaller fragment, it is released into fluid phase, and the BB, which is a larger fragment, it remains attached to the surface of pathogen. and as a result of these interaction, a complex is formed which is C3BB, and this complex is the C3 convertase of alternative pathway. Alternative pathway. And these C3BBB like C3 convertase of lectin in classical pathway is going to cleave the next component, that is C3 component of the complement system. Clear? So it was all about the lectin pathway of complement activation and the alternative pathway of complement activation. And next, King Tong Ping Pong. Sumra chale. Lekada ho sumya ra bara ne sera po shuma.